Have your attention, please.
Hi.
Darcy, looking at you. Thank you. We're all here today. We've got. We just wanted to introduce our very good friend Gall.
Yeah, but
Yeah, I don't see him here.
Not here right now.
That's usual. He always shows up late.
I know, that's typical, guys. Gall's always late.
So,
Let's just start with the more fun stuff,
Yeah,
which
that's a
isn't
good
Gall
idea.
actually. It's his dog. Gall has a dog. You might have seen her at
Wonderful dog.
TGIF. Her name's Belle. She's a cutie.
They have a good time together. Belle and Gall, they're wonderful.
Yeah, they share a bed. Super cute.
Here's Belle in a wedding veil.
Yeah.
Adorable.
Here's Belle being sassy.
And just one more.
Winter Belle.
Yeah.
Belle in winter.
And the other wonderful part about Gall is his wife.
Little.
Who you can see their save
This is
the
their
date.
save the date.
In
Super
the
cute.
wedding.
They're both black belts in karate.
Mhm.
There's Lital. She's also my shopping buddy. You guys need a shopping buddy? Lital will shop for hours. Like she will go and go and go. And it's great.
So we also noticed in our research that Gall is also a black belt and is proficient in knife fighting.
Yeah.
Although he does have to show up to the match on time, which can be difficult.
Yeah.
We figured that since he was going to be late, he's probably actually just working on making slides for the presentation today.
Yeah, that's probably why he's late. You don't see him here.
So we wanted to make a few slides that he could use just in his presentation. If he has some kind of fluorescence.
Yeah, just slip these in if
just-
you're having little holes in your presentation ball.
Just you can pepper these in. You got some kind of strand line that you
Maybe
need.
correlation.
Just throw these in.
Just throw them in.
Throw these into your presentation.
Just whatever you want.
Yeah,
That's
great. So I guess we don't really have much more to say. We just got to fill up some time until he gets.
Yeah.
Wait.
Is Gall here?
Yeah. Gall.
Oh!
When did
my
you
God,
get
there
here?
you are. Gall, we couldn't even notice you because you don't have your dog.
Oh my God. You gotta let us know. And I'm glad you're here because this
We
is
wanted
the most
to
important
sort of
part.
highlight one of your hidden talents that you have yet to show MCO, which is this little gem. All right. Yeah, use them, throw them in.
What is the title say?
Yeah, best of luck. But you didn't leave them.
Look it down. I can't.
Thanks, guys.
Here, take them all. We'll just put them up here. Okay, I'm gonna keep this though, so.
Thanks, guys, for these kind words. So. Okay. So this is the time of my talk. Too much chewing up will give you holes in your brain. Let's just get to it. So, we're all scientists, right? And what do scientists like the most? Well, we like grant money. We like free food, especially pizza and beer. We like getting sent to conferences because that's free money and food. Best of both worlds. But when we really look at ourselves, what's the thing we really value the most? That's right. Our brains. We all use our brains a lot, and we probably want to have long and successful careers using our brains. So that's a brain. And there's a problem though. There's this disease out there. Sorry. Things. that's threatening to take these brains we love so much and put big holes in them. And so this is a general illustration of the process. And yeah. So that disease is Alzheimer's disease. And when we look at what happens in the brain during Alzheimer's disease, we see holes, not like these, but similar. And we see these plaques. These are called amyloid plaques. And scientists that look at these plaques, made the obvious connection between cognitive decline, memory loss, and so forth in Alzheimer's and these plaques. So what are these plaques? Well, the plaques are made from amyloid beta. And amyloid beta is a protein that's derived from this amyloid precursor protein over here. It's a transmembranous protein. It needs to be cleaved twice before it becomes amyloid beta, once by beta secretase and then again by gamma secretase. Then we get these peptides, which are about 36 to 43 amino acids long. And generally, the ones that are correlated with the disease are 42 amino acids long. Probably most of you heard of amyloid beta 42. It's 42 amino acids long. So these peptides then can aggregate into oligomers, and the oligomers can then aggregate into the plaques we just saw. So, but we've been trying to get rid of these plaques for like 20 years or something now, and whenever we have some success getting rid of them, Alzheimer's disease is still like a cold in the brain. People are still having cognitive decline and so forth. So what's going on? Well, we know that the brain is basically a web of synapses, of connections between neurons. And this is the general illustration of how of how neurons synapse. If we zoom in over here, we can see there's a little gap between the neurons, between the presynaptic neuron and the postsynaptic neuron, and this is where the signal goes through in the form of neuron transmitters. Now, synapses naturally want to make these, neurons naturally want to make these synapses. And so some of the time during brain development, the brain needs to kind of cut some of them off so that the brain can develop properly. And this is achieved by a process called synaptic pruning. Now, in a paper that preceded the one I'm going to be discussing, they discovered that synaptic pruning is done by microglia. This is a general sketch. So there are these phagocytes of the brain, right? They eat up intruders, and like I said, they help with the synaptic pruning process. Now, the following is an artist's rendition of what happens during synaptic pruning. Yeah, I hope that is educational. The other thing they discovered in the paper I mentioned is that this process is mediated by the complement system. Now, the complement system is a very complex system. It's part of the immune system, and it's comprised of like 30 or something proteins. I'm not going to talk about all of them, obviously. We just need, for the context of this paper, the following two. So the first one, just going to take a protein out of the complement system here. First one is a C1q, and C1q kind of just hangs around the synapse. It's followed by C3, so you kind of get the naming regime here. All the complement proteins have C's in the names. C3 follows C1q, and these microglia have this little receptor here, which you can see. That's a receptor for C3. So when C3 comes to the synapse, the microglia can follow and basically engulf the synapse. All right. So in Alzheimer's disease, one of the things that are a better marker for the disease progression is synapse loss. And we see synapse loss here, synaptic pruning here. If this kind of process goes awry, it probably has bad effects on the brain, right? So this is where the paper I'm going to be discussing comes in. Title of the paper is Complement and Microglia Mediate Early Synapse Loss in Alzheimer's Mouse Model. It was published last year in Science out of Beth Stevens' lab in the medical school here at Harvard. Okay. So like the title suggests, the paper uses a mouse model of Alzheimer's disease. They basically took the amyloid precursor protein gene, human APP, from familial Alzheimer's disease carrier or early onset Alzheimer's disease. This is an allele that has 2 mutations, and we're not really sure what they do, but they're correlated with the disease. So they inserted this gene into mice and named them Jclub. So first we want to know how good is our model. So the first question that the researchers asked is, does synapse loss occur in J20 mice? So they took a slice of the brain, so basically neurons, right? And they tagged presynaptic and postsynaptic neurons. Here we can see PSD95, or postsynaptic density protein 95, and synaptophysin, which is presynaptic protein. And obviously in the microscopy, you don't really see the neurons. You just see a bunch of dots. But you can kind of imagine that you can count dots, first of all. And you can count how many of them exist in the same vicinity or in the same area. And so you can kind of get a sense of how well your synapses are doing. So when we look at the quantification of this, we see synaptophysin is pretty much unaffected, but both PSD95 and the quantification of colocalization is reduced by about a half. This is a relative plot, so it's it's a percent of how much of whatever we saw in the wild type. This is the general type of plot we're going to be showing throughout the presentation. All right. So now we know synapse loss occurs in J20 mice, and this was also confirmed by other proteins with the same assay, and also by electron microscopy. So we know that we have a pretty good model of Alzheimer's disease, and next we want to look at our first factor in the synaptic pruning process we saw before, which is C1q. So we asked, how does C1q behave in J20 brains? So let's go back to our brain slice here. And when we tag C1q and measure how much of it there is, we see that in the wild type, we see much less C1q than in the J20 brains. Okay? So C1q is upregulated in the J20 brain. Next, we want to see if C1q hangs around the synapse like we saw in the synaptic pruning. So we do the same, pretty much the same thing. We do colocalization. We look at the dots and see how many of them kind of correlate to the same general area. So we do that, and we see that in a J20 brain, colocalization is pretty much double. All right. So the conclusion is that C1q is upregulated and localizes the synapses in J20, similar to what we see in synaptic pruning. Just if anyone's confused, a little summary before we go on. We have a strain of mice that exhibits synapse loss in the pre-plaque brains. I haven't mentioned this, but all these mice were harvested about 3 to 4 months old, and this is way before plaques can form. So plaques do form in this strain, but much later. C1q is upregulated in J20 brains, which you just saw, and it co-localizes with synapses. Okay. So we know the complement system behaves somewhat similarly to what we see in synaptic pruning, and now we want to see what happens to amyloid beta. And that's because the main difference between our strain and wild type is the amyloid beta gene, right? The amyloid precursor protein. So the next question we're going to ask is, does amyloid beta cause C1q upregulation in J20? So we see wild type versus J20 brains. And we already know that if you tag C1q, you see a lot more in the J20 frame. When we then tag amyloid beta, we see an upregulation in the J20. However, when we block amyloid beta, we see that C1q upregulation goes away. And this inhibition was achieved by a compound called compound E. It's basically a gamma secretase inhibitor. So if you remember the one that cleaved the second bit, so we see that C1q upregulation is dependent on amyloid beta. But which amyloid beta? So we saw we have several types. Like we have plaques. We know that's not it because these brains don't have plaques yet. But we still have monomeric and oligomeric amyloid beta. So, which amyloid beta form affects C1q regulation? If we compare an empty injection, so this is injection to wild-type mice. If we compare an empty injection versus the monomeric versus the oligomeric, we see that only the oligomeric form upregulates C1q. Okay. So now I'm hoping you all pretty much get the general gist of the method of the paper. We tag proteins, we try to look at them in the context of synapses, and we see what's upregulated, what happens where, and so forth. And so I want to summarize the final experiments with a few animations. Let me just set it up here. So we have our wild-type brain. We can tag synapses, same as before, and we can tag C1q. This is like general shape that we saw in the introduction. And so when we inject the A beta oligomers that we saw before, we're doing the C1q upregulation. We see that C1q localizes synapses. And this causes synapse loss. Okay, now when you knock out C1q, let's knock it out. We can do the same injections and nothing happens to the synapses. Okay? Now we haven't talked about the microglia so far, so what's up with them? So you remember the microglia are these little Pac-Mans here. And so when you do the same injection, you see that the microglia comes along and engulfs the synapse. However, you remember this complement protein receptor we have over here. If we knock it out and do the same injection again, you're probably going to tag this time. Well, nothing happens to the synapses again. And so we can conclude what we learned in the paper. First, developmental synaptic pruning pathways might be involved in Alzheimer's. I'm saying might because we had a mouse model of Alzheimer's and we had an early onset model, not the late onset model. The late onset model, the late onset disease is correlated with different genes. These pathways are activated before plaque formation and cause synapse loss. And like I mentioned, synapse loss is a much better marker. It makes sense, right? If you lose synapses, you lose brain function. The general pathway we described was that A beta oligomers cause the complement system, which is basically C1q followed by C3, to recruit the microglia to then engulf the synapse. A few future directions that this work might take. First, we've seen a few potential therapeutic targets. So you can imagine that if this process is mainly involving development, knocking it out might not be such a bad thing. Blocking it, or there's many ways to block this pathway, and so it might be not such a bad idea to try and block it and see what happens to the disease. The other thing that could be done is a genome-wide association study, which you could look at the general population, see the ones that have the mutant APP gene, and see if complement mutations inhibit the disease. Finally, a little criticism. First, you remember I showed that they show that the microglia activity is dependent on the complement by knocking out the complement receptor, CR3, on the microglia. And I was a little bit confused by why they just didn't do the same experiment again with C1q and see if it has the same effect, because CR3, the complement receptor on the microglia, is a microglial protein. So knocking it out might have an effect that confuses the system. The other thing is that it would have been nice to see some behavioral assays for injected wild type versus complement knockout mice. This is maybe something that they're working on. So finally, I'd like to thank my coaches, Catherine, Susan, and Ikun, and you guys for listening, and I'm not any questions.
So they sacrificed the mice pre-plaques, right? Did those mice show cognitive deficits? Like, did they test them in any way?
Yeah, so I mentioned, I don't think they did. Not that I'm aware of.
Do you know, like, is it a well-studied mouse model? Like,
They have borrowed it from previous studies. It has a couple of years on it. I'm not sure if anyone actually done the behavioral assays. But I think that in the stage that they're doing the harvesting, you probably wouldn't see too much behavioral changes. You have to wait a little longer. Yeah.
So, if I understood right, the hypothesis is that the oligomeric A beta directly upregulates C1q, which translates to the microglia pruning and things like that. Right?
We don't know if it's direct either.
Or rather, it is correlated with.
Yeah.
Right. So, I guess my question is, in that vein, did they attempt to just directly upregulate some artificially just C1q without relying on A beta, right? Because that would at least tell you, is there a one-way path from upregulation of C1q all the way down? Right? Because right now it's not yet clear if it's just a correlation or C1q upregulation is a direct causative agent.
I'm not aware that they did that, but even if you did, I'm not sure it would tell you anything, because you can imagine that amyloid beta not only upregulates C1q but also changes some of its function. But I'm not aware that they did.
Thank you.
Yeah.
Do you know offhand, so pruning is a signal for the pruning lack of activity? And does A beta actually dampen synapse activity? Like, could that be the commonality that might bring complement to both cases?
I guess it's an interesting idea, but I don't think that that they know what initiates the synaptic pruning. The paper that I mentioned that preceded this, so that's 2016, that one was 2012 maybe. So it's pretty early. I'm not sure that they know what initiates it. They just know that it happens, and when you block that process, the brain develops poorly. So function isn't impaired in the adult. Yeah.
So in the wild-type situation with age, it's known that you have a less amount of synapses being formed than normal, right? So do you expect that the effect here is just because you always have the same rate of synaptic destruction, and you just lose the rate of synaptic de novo formation? Or do you think it's something else that is specific to these mutant alleles that just accelerate them?
So what I understood from the paper that the driving factor here is synapse loss. So it's not that synapses are formed in a decreased rate. Is that synapses that already exist are getting engulfed.
Yeah, but as you get older,
Yeah.
it is known that you have less
Mm-hmm.
synapse formation activity. So what I'm asking is this then what determines that you're going to have the pathology, just because you're making less of them as compared to when you are younger? Or do you think it can be something else?
I don't know. Like from what they describe here, it seems that it's loss of existing synapses and not inhibition of synapse formation. But could be something that exacerbates the disease. But the actual human disease, the early onset human disease, is I think usually start seeing symptoms around 50. So I don't know if in that age you start seeing this.
So I still have some time.
Yeah.
Do you think this is a common mechanism for other neurodegenerative diseases?
So yeah, in the paper, or in some of the papers out of the Stevens lab, the first thing that they saw was that this process is involved in some, I forgot exactly which disease it was, but it was some neurodegenerative disease of the optical system. And that's kind of what drove which is the author, to kind of look at Alzheimer's as well. So yes. Okay, thanks everyone.